Hi everyone and welcome back to Advanced Higher Biology. In this video we are going to finally finish off Unit 1 Cells and Proteins by concluding the fifth key area, protein control of cell division, in Part D, control of programmed cell death or apoptosis. So again, in this key area we've had a look at how the cell cycle is controlled by proteins, what the checkpoints are, what happens if certain conditions are not met, and one of the final things we're going to discuss is what apoptosis is and how it can be activated. So if you've not came across this before, apoptosis is essentially programmed cell death. It's the cell destruction or self-destruction of that cell, and it all sounds very, very dramatic. So in apoptosis, that can be triggered by death signals that are either external out of the cell or internal within the cell. And the main part of this is knowing examples and comparing both. So in terms of external death signals, a good example of this is a lymphocyte go across and breaking down some form of pathogen. So an external death signal molecule binds to the surface receptor proteins of the cell. So remember, it's on the outside, and it's going to trigger a protein cascade within the cytoplasm of that cell, and again, leading to the destruction of that cell. In a similar form, internal death signals also lead to the destruction of that cell, but obviously, as the name suggests, it is internally. So a good example of this is when we looked at P53 in the previous subkey area, when we looked at the G2 checkpoint, checking what's going on with DNA replication, and if there was damage to DNA, then it's going to activate the P53 protein, and that then, of course, leads to cell destruction as well. So again, very clear, external proteins outside, or external death signals rather, outside the cell, causing destruction, internal within. But they both have the same end result, and they both result in the activation of a type of enzyme called caspase. So these caspases are going to be activated within the cell, and that leads to a pathway leading to that self-destruction, okay, of that cell. Now, in terms of apoptosis, that is used to destroy cells that we don't want or cells that are abnormal in some way, but it can also be used during development, during evolution as well, or metamorphosis, when we want to remove cells that are no longer required. So a good example, you might see some pictures of the development of hands, or there's some metamorphosis in frogs and things, where if you have these big webbed hands and you want to remove the sort of webbed area between so you have functional fingers, those cells can be destroyed through apoptosis to basically remove them, and then you're left with the parts of the cell that you want. And what can also happen as well is cells may initiate apoptosis if there's an absence of growth factors. So if there's no growth factors being added, then the cell may just decide, right, well, we're not needing this anymore, we'll just destroy it through apoptosis. So that's it. That's all you need for this sub-key area. It's very, very quick. It's very small. Like I said, just be aware of what apoptosis is, what is a use for it, and know the differences and examples between internal and external signals that lead to apoptosis and this dramatic destruction of a cell. So, like I said, that is the end of Unit 1. That's these videos finally done. I know it's taken a little while, and I appreciate all the messages that you've all sent in and how many people have found these useful. Apologies, it's taken a little while to get to this stage. Very quickly, just to explain what the future of this channel is going to be, because I know quite a lot of you are using this for your advanced higher. Essentially, what we're going to do is you should be aware that we've done Unit 1, and there's also Unit 2 and 3 of Advanced Higher. Unit 3 itself is actually more of a half unit. It's only 3 key areas. It's quite quick, it's quite straightforward. And certainly in my school, we actually start with Unit 3 because it gives you a bit of a grounding in experimental design, scientific method, and it's very useful for planning out your advanced higher projects. Even if you're carrying them out or you're writing them up just now, it's quite a good way of looking at your process and the actual process of science itself. So it's not really the biological knowledge as such, it's more the principles and processes of science and research as well. Now, it is quite quick. Like I said, in my school, we tend to do it first and then Unit 1. So what I'm going to do is we're going to start off the next series of videos on Unit 3 and try and get that part out of the way as well. I do know of quite a lot of schools that also teach it in this, albeit very annoying and non-logical format. But what it means is that going into prelims, hopefully it kind of covers those areas for you and then we can get on with Unit 2 and get the whole area done. So if you're going through the process of Unit 1 then Unit 2, apologies, there is going to be a brief halt where we'll do Unit 3 and then go back to Unit 2, which is an amazing unit, so it should be worth the wait, hopefully. But we are going to go into Unit 3 next. So hopefully see you there for those videos. Hopefully you find them useful and that you've enjoyed the Unit 1 questions as well. Okay, so very best of luck for those of you who have prelims coming up. And again, hope you find these useful. Thanks for getting in touch, and I'll see you soon for Unit 3. Bye for now.